Question number 151. Select the term which is not associated with moit. What is moit? Moit is multiple ovulation, embryo transfer. It is a technique in which embryos are transferred from their genetic mother to surrogate mother. In moit, the fertilization occurs inside female's body. So they exhibit what? They exhibit in vivo fertilization. Now the question asks for the term which is not associated with moit, and the very first option, embryo transfer. Embryo transfer is associated with this, so this cannot be a correct answer. Super ovulation is associated with this, so this cannot be a correct answer. In vitro fertilization, as moit involves in vivo fertilization, so this is an incorrect. So this term is not associated with moit and is a correct answer to our question. The last option is artificial insemination, and as it involves artificial insemination of sperms into the genetic mother followed by the fertilization of sperm and ova, so this term is also associated with moit, and so this is an incorrect option. So the correct answer for this question is option number 3. Let's discuss our next question. Question number 152. During detection of cancer, technique that uses ionizing rays to generate a three-dimensional image of internal organs of a patient is, and the very first option given is autoradiography, and this is an incorrect option because autoradiography, it is a method which allows the detection of radioactive isotopes within a biological sample. So this is an incorrect option. The second option given is CT scan, and this is a correct answer to our question because CT scan or computed tomography and X-rays, they uses ionizing rays for the detection of cancer. Now the next option given is MRI. MRI is magnetic resonance imaging, which use strong magnetic field and non-ionizing rays to detect cancer. So this cannot be a correct answer. Our last option given is biopsy, and biopsy as well as histological biopsy as well as histopathological studies are the methods in which a piece of suspected tissue cut into thin sections and then stained and is analyzed under microscope. So this cannot be a correct answer. So the correct answer for this question is option number 2. Let's discuss our next question. Question number 153. Read the following 4 statements about RNAi technique and select the incorrect statement. So this question is based on RNA interference technique. And what is this technique is all about? It's a post-transcriptional mRNA silencing technique which prevents the expression of specific genes. So the very first option given is, it is a post-translational mRNA silencing technique. This is an incorrect statement and is a correct answer to our question because it's not a post-translational but a post-transcriptional mRNA silencing technique. The next option given is, using agrobacterium, nematode-specific genes were introduced into tobacco plant. And this is a correct statement because these nematode-specific genes were introduced into tobacco plant where both sense as well as antisense RNA are produced. These RNA being complementary to each other form a double-stranded RNA that initiates RNA interference and thus silencing the specific RNA of the parasite. As a result, the parasite cannot survive in the host plant. So this is a correct statement. The third statement is, RNA interference is a mechanism of cellular defense in eukaryotes, and this is a correct statement because RNA interference is a method of cellular defense present in all eukaryotes. The last statement is, formation of double-stranded RNA in host plant triggers RNA interference resulting in protection against nematode infestation, and this is also a correct statement, so cannot be a correct answer to our question. So the correct answer to this question is option number 1. Let's discuss our next question. Question number 154. Which of the following is not a feature of plasmid PBR322? Now, what are plasmids? Plasmids are extrachromosomal, closed, circular, and autonomously replicating DNA. And generally, they do not integrate into the chromosomal DNA. The very first option given is double-stranded circular DNA, and as this feature is associated with plasmids, so this cannot be a correct answer. The next option is autonomous replication, and as these plasmids exhibit the autonomous replication, this cannot be a correct answer. The third option given is integrates with chromosomal DNA in host cell, and this is a feature which is not associated with plasmid PBR322, so this is a correct answer to our question because generally these plasmids do not integrate into the chromosomal DNA in host cell. The last option given is absence of histones, and as these plasmids lack histones which are generally associated with the chromosomal DNA of the host cell, so this is an incorrect option. So the correct answer for this question is option number 3. Let's discuss our next question. Question number 155. Select the incorrect match with respect to the cell and cell wall or membrane degrading enzymes. And as we know that the degrading activity of the enzymes is based on the composition of the cell wall or cell membrane. So according to it, in the very first option, for yeast we use chitinase, and this is a correct match, so cannot be the correct answer to our question. The second option given is bacteria, and the enzyme is lysozyme, and as it is a correct match, so this cannot be a correct answer because lysozyme is used to degrade the cell wall of bacteria. The third option given is plant cell, and the enzyme used is cellulase, and as the cell wall of plants contains cellulose, so the cellulase enzyme is used to degrade the cell wall of plant cell, so this cannot be a correct answer as it is a correct match. And the last option given is animal cell, and the enzyme used is RNase, and as it's an incorrect match, so this is a correct answer to our question because this RNase will hydrolyze the RNA which is present inside the cell and not on the cell surface or cell membrane. So the correct answer for this question is option number 4. Let's discuss our next question.